There was a PA that I saw work with kids in a pediatric setting, which makes sense. That she would say that, let's see your ear balls, and they would all just panic and laugh and distract them, and she could get in there. And I thought that was really nice. I like saying ear balls. Oh my glasses. Okay, do you remember Drumline? Did you see Drumline? Because if you haven't, you need to see it. Oh my God, I love that movie. What is it about a drumline though? Do you have that reaction to a drumline? I mean, like, it's like fireworks, you want to weep. I mean, but there's something about it because you can feel it, you know? You see it.
No, my friend was sitting here.
Anyway. I was in band. I know you're surprised. So anyway, some of y'all are in band. So anyway, auditory system, noise. I thought of drumline because you're perceiving sound waves, which is like light waves, but slower and not as pretty. There's sound, and it seems easier. But I think that it's so weird once you try looking at just tapping, the little ossicles tapping on an oval window, and somehow you get sound out of that. That's pretty weird. But because an object vibrates, it creates sound waves. And that percussion or movement of air is what translates to your ears. So really, it's something that's moving, it makes a noise and you can hear it. And so with that, let's enjoy the ear balls, shall we? Oh, do you just love Nick Cannon? Very stupid Mariah, I'm so glad they were good. I'm sorry, she's so ugly. She's probably not that stupid.
Oh my gosh.
Your ears. We don't have movable ears really like other mammals, but wouldn't that be cute? And you can listen to someone. Does your dog talk to you like that? Calm down, boy. All right, so you have the outer ear, the middle ear, and the inner ear. Let's look at each section, shall we? And we send that information to the thalamus, to the, why do I have primary visual cortex? Oh, is the auditory cortex on there? I can't see it. Yeah, it is. Of course it is. I put that slide up there, it makes sense. Okay, so anyway, auricle, and you have this helix and this earlobe, and they're kind of oriented forward so you can gather sound waves. In an anterior sense, the ears are bilateral, and so you do have information being sent to each hemisphere so that you can lateralize sound like we do with our vision as well, of course. In external auditory meatus, covered with epithelium that secretes cerumen, which is the ear wax. It's kind of a modified sweat gland, and you make earwax. And that epithelium grows in a sort of spiral sense from the interior of that canal outward. So stuff should be naturally moving outward. You know, they always say don't stick anything in there, but when you'll have a kid someday, you're going to know. You're going to be cleaning and gouging in there as much as you can. But if you have your otoscope, you can put it in there. Let's see, what could I say about the external auditory meatus? There's a few little hairs in there. The outer ear ends at the eardrum. So there's your auricle. You don't have to know all of those curvatures, but you know, it's with a purpose. It kind of has a Snell sort of, what's this called?
Shell.
Yes. Whorl. A whorl. Whorl. Excuse me. And then the external auditory canal with little chunks of earwax in there for realism. And then you know that you're kind of buried in this bone. Do you remember what this bone is?
What?
Yeah. It is. That's good. You had a little hole there where that was coming through, and all that inner air is really just in this, all this mess of bone there. So anyway, is this what I want to say about this? It seems like I had something more to say. No. Oh, here's one. The earwax, it's a little bit acidic. It's a little bit antibacterial property to it. And part of it is, because you're always sloughing dead cells all the time. Part of it's going to trap dead cells and keep it moving, but it's kind of a conveyor of stuff out there so that you can keep that canal patent, because you have to be able to have sound waves come through there. If there is lack of patency or openness of that tubing, then you have conductive hearing loss, meaning the waves aren't conducted to the place where they could be heard. And then you end the outer ear at the tympanic membrane and begin the middle ear, which is an air-filled space. And parts of the middle ear is the tympanic membrane and the auditory canal, or eustachian tube. The epithelium of the eustachian tube is continuous, and it is open to the nasopharynx, or that early part of the throat. And I show you this picture because, the big pediatric thing that you experience is the middle ear infections and otitis media, where fluids are built into this middle ear, or air-filled space. Fluids are there, and then that decreases the ability of the tympanic membrane to vibrate, so that decreases hearing. And in children, the auditory canal or eustachian tube is more horizontal just because they're little, and then you have more incline in that tube as the head gets bigger. Although there's some big-headed kids out there. And so you would have draining there. So this tube is naturally closed and will pop open when you yawn or chew in an effort to equilibrate pressures, the atmospheric pressure with that middle ear pressure. If the atmospheric pressure and the middle ear pressure are the same, then the tympanic membrane is freely mobile. And so that's why that closed feeling or stuffiness of your ears is when the pressures aren't equal. And really, you can't hear as well on that side. The structures or the anatomy of the tympanic membrane, so you'll look at the fundus with the otoscope, you'll look at the tympanic membrane with the, what did I just say? You'll look at the fundus with the ophthalmoscope, and you'll look at the eardrum with the otoscope. And so you'll really, this you can see. You'll love this because you can really see it. And my kid was like 3. I looked at her eardrums constantly. It was horrible for her. I can still just come at her and she'll go like this. Horrible. I looked at her because they get a lot of ear infections. So they're terrible. Just, it's so good you don't have one of those. Love the eardrum. It has this kind of tinted cone shape. It sort of tints deep. It tints deep or toward the middle or midline. And you can see through it because it's thin, and you can actually make, you see the inner ear structures through there. But you have this umbo where you can actually see the malleolus, where the malleolus is, or the little ossicle laying against the tympanic membrane that would receive the vibration. So you can actually see that. There will be a cone of light because your otoscope is lighted, because otherwise it's dark in there. And you'll see that reflected and always in the same place. So if that's moved a little bit, then something isn't quite right. You can sometimes see an air fluid line, which is always really exciting, and bubbles. It's always really exciting because you can actually visualize. This is one of the things you don't really get to do so much with eyes, although there's a ton of stuff you can diagnose with by looking at eyes, from color of the sclera to injection of the conjunctiva and lots of things, but this you can actually see and diagnose something, so that's kind of nice. Let's see, other structures, you really call the 12 o'clock portion is the pars flaccida. It's more mobile, and then the pars tensa is really the remainder of it. And there's an annularis or ring around the corner, around the perimeter of it. And so those are sort of the landmarks of the tympanic membrane, and that's really the thing that receives the sound waves and begins the vibration. And somehow that creates all of this variety that you can listen to music and hear all the different parts of it. And that's, again, it's miraculous. It's like life is miraculous. And it's really exciting that you get to study it. You'll forget that and you'll hate yourself. But if you can, always build in a little bit of time where you can just sit and think about the fact that this is really an exciting thing to get to do, is learn about this stuff. And further, go get people feeling better. Or, or worse. Just do well on that exam. But you get lost in the burden of the details, and you don't want to keep going. And so I think it's really important to spend that little bit of time, build it in, meditating on that it's a miraculous thing that you're getting to study, and maybe that'll keep you going, or maybe it'll keep you from doing something bad to yourself. I don't know. Sorry. The dark current came. I'm nearly over PA school. No, I'm really not. Okay, so middle ear, from tympanic membrane to really the oval window. So this fluid-filled space, the tympanic membrane. See how it cones in? Not quite to that extent. And it is in contact with the three ossicles. I know you like. Do you just love them? Do you love these little ossicles? These tiny little bones. I mean, I don't know why. They're adorable. That's why. There's the malleus, the incus, and the stapes. And look at the malleus and incus. What they're really doing is they're kind of like a lever system, the way they're attached. You're not just knocking on one, knocking on another. The way that they function is to amplify the sound, because you have this large tympanic membrane, and you're trying to conduct that vibrational energy over to a very small place where the stapes is wrapping on that little oval window. That's where you're trying to transmit that information. So you really have to amplify it from the tympanic membrane to this oval window. And it's the function of the ossicles to really do that. And it's especially this head of the malleus, where it articulates with the incus, and that lever action really helps to increase the vibrational quality of what's coming in. Let's see, I believe you have to, maybe it's a 20 times magnification. Oh, I'll get to it later, but you have to overcome the fact that that oval window is really little, and the inner ear where you're trying to get that vibrational information is fluid filled, so the fluid itself resists movement, even though that's what you're trying to create. There's more inhibition really than there is excitation, and so you have to overcome all of that onto this small area where the stapes is moving. So anyway, the ossicles will amplify the sound that's received from the vibration at the level of the tympanic membrane and conduct that to the inner ear. Let's see. That manubrium you could see, that was at the umbo that you could see on otoscopic exam on the tympanic membrane. That's all. And then there's some teeny little muscles. Precious, aren't they precious? The tensor tympani, or tympana. Who is not from Oklahoma? We say tympana. And I insist that you say the same. The tensor tympani is attached to the malleus, and it can contract and pull the malleus away from the tympanic membrane, so that's a way of making you not be able to transmit sound. So that's a reflexive action to very loud sounds. There's also the stapedius. So let's see, I can't see that. Where's this? Oh, the stapedius muscle there attached to the stapes, which is a cute name. And it will contract and pull the stapes away from the oval window. And these can both happen, and it just really stops sound from being transmitted. For a loud sound, it's reflexive. And that's the tympanic reflex, and I think I just have, I don't have that word spelled right on your slide there. I think I made a correction there. So anyway, it's called the tympanic reflex. But it's a little slow, and so if a gunshot happens next to your ear, hopefully that's just not a scenario that will happen. But you can't really help that. But if it's a loud, repetitive noise, then you can kind of be protective to the ear by that tympanic reflex. And those little muscles, all those little bones, are so precious. Your little ear balls. Okay, now, the inner ear is really this, I wish I could move that off of that word that's bothering me. It's the space and the membrane of the inner ear. So the bony labyrinth is really the very worm-like, torturous pathway that is seen in that temporal bone. So this is really all just surrounded by bone. And then the membranous labyrinth then is the stuff that's inside there, or really the inner ear. There are two fluids, perilymph feels that space, and endolymph would fill the membranes. But it's going to be a little more complicated than that. But there is a cochlea, which is anterior to the vestibule, and the semicircular canal. Semicircular canals would then be posterior. And you know all these guys, don't you? That little cochlea smell like with whorls. It's the size, it's half the size of a split pea. I picture, that's not something you ever dissect, is the inner ear, because you would destroy it by the time you got through all of that bone. But, like everything else, I pictured a lot bigger than it really is, which works out sometimes. But the cochlea is just tiny, and it does all this wondrous work. So, anyway, let me see. No, sphincter. And then look here. The vestibulocochlear nerve, which one is that? Number 8. Yes, it is. Has those two sections, the vestibular, what am I trying to say? Vestibular nerves and the cochlear nerves. And they're coming from the inner ear because one is hearing and one is the balance and perception of movement. So cochlea is hearing. And so those fibers come from each of those places here in the inner ear, coalesce, and go to the auditory cortex and elsewhere. I'm getting late and I'm having less than remembering. Okay, yeah, this is going to get complicated. All right, let's go into the cochlea. Okay. The cochlea is this 2.5 turn spiral of membrane. There's some more membrane dividing it in here inside there. So, you're just typing away. So I like this picture. If you could see the cochlea in cross section, because you get lost in these little pictures, but you just have to remember that it's this. And so if you cut it in half, it would just be so much like a snail. Oh, my stomach just growled. It's getting ready to get real loud up in here. It is not a loud growler. Okay. So there are 3 distinct chambers within this cochlea. And we'll start with the middle one, because it's actually called the cochlear duct. It's the wedge-shaped one. It's also called the scala media, where scala is referring to chamber, which makes me think of scallops. Cue the growl. So look at the scala media there, and it is kind of wedge-shaped. So let's stick with the scala media. There is a stria vascularis forming the lateral wall of the cochlear duct, and it's vascular, and it will produce the endolumph that is located or inside this cochlear duct. Endolumph, so a fluid on the interior of a duct. Endolumph is found in the scala media. And then some other parts that are really important within the scala media are really the hearing parts. And so the organ of Corti, see, I did not say corti. The organ of Corti, and sitting kind of resting on top of it is the tectorial membrane. Now, I'm saying all this here, but we're going to get to each part. But I think that's the business part of this, and so I want to spend some time discussing that. So, organ of Corti, the little hair cells, the little receptor cells, and the tectorial membrane that forms their roof. Now, a couple of other membranes then that form the rest of the scala media. So, lateral wall of the scala media is the stria vascularis. The roof of it is that Reisner membrane. So you see Reisner membrane, and I put MB for membrane, separates the cochlear duct from the vestibular canal. Okay, now this sucks, because these chambers have more than one name, like everything does. So cochlear duct is scala media. The superior chamber on top of that is scala vestibuli, and we can also call that the vestibular canal. Ugh. And then the floor of the scala media is the basilar membrane. That's really an important thing. We'll talk a lot about that. And it will separate that cochlear duct or media, scala media, from the lower chamber, which is scala tympani. Are you guys even hearing me anymore? Because this is a lot. I mean, I get it. It's hard. It's hard, isn't it, just to stay like plugged in. But it's so darn interesting. So let's keep going. And then look there at the spiral ganglion. Those are, it's called a spiral ganglion because these are the nerve fibers that sort of go around this, or don't sort of go around, they spiral around, hence called spiral, around the cochlear duct, or the way that the cochlea is shaped. And they will transmit the information to along the cochlear part of that cranial nerve number 8. So let's go back and look. Scala vestibuli, scala media, scala tympani. The membranes that separate are the Reisner membrane, and that's not very important. The basilar membrane we're going to discuss much more. And then the organ of Corti, which is in scala media, and that's really the receptive thing that will generate the impulse that will be sent along the nerves. Oh, where am I going? Right here. And so there's a few pictures of that. Okay. Now, basilar membrane. We're going to just take each of those chunks in from the cochlear duct or scala media, that wedge, the business portion of the cochlea. We're going to take each chunk. So here is the basilar membrane. And it's one of the things that gets moving when we translate tympanic membrane vibrations through the ossicles to that oval window, and then that gets conducted along the cochlear fluids, and it's really the basilar membrane that starts rocking. Now it has this specific sort of shape where, let me make sure I've got this, I'm telling you this right. Basilar membrane near the, so it is the opposite of what it shows here. Okay, that's right. So see this little, let me get there. So see this little, what is this called? that little picture with that. Okay, my words are gone. Hold on. The basilar membrane runs along the length of the cochlear duct, and it's found as the floor of the scala media. It has a particular shape such that near the oval window, which originally receives the vibrations, it's wide and thick. No, it's not. Here, I said it wrong. The basilar membrane near the oval window is thick, but it's narrow. It's pretty floppy right there. And then as it goes along, if you could stretch it out, it gets thinner and wider. And all that really means is that it's able to receive a vibration in a particular way. The shape doesn't matter so much. And it has fibers running horizontally across its length, not longitudinally, but horizontally across its length. And these fibers will vibrate maximally at a certain resonance. So a sound wave will travel through, and these fibers in a particular spot will really respond to that sound wave and begin to vibrate. And what they will do is they'll create mechanical distortion of cells up there in the organ of Corti, which will then submit or send a signal to the brain. So here's what I want to say. Like light caused a conformational change in rhodopsin or in one of the photoopsins, and that became then an electrical stimulus. What we have to do here is take a sound wave and create a deformation that will ultimately result in an electrical stimulus. And it's the basilar membrane that begins that. So if we can look at it here, it's spread out in the picture over here. I can straighten it out. And you can see along there where it likes to receive what resonance of wave. So near the oval window, and that's where the stapes is knocking against that fluid. Those are going to be high frequency sounds because they're doing this, they have a higher frequency. And somewhere in the middle a little bit lower, and at the very end lowest. And so you can just conduct that in this other picture where you can see it still coiled up. The different hertz, which is the cycle that correlates to the frequency of the sound. 20,000 hertz at the beginning, and you get all the way down to the end, and you see you can hear at 200 hertz. So that is how you understand where in that sound wave, where that sound wave is, or the frequency of that sound wave, because the basilar membrane is tuned to a particular frequency. When I'm hearing this come out, it doesn't sound very much like it's making sense. Is it the time of day, or is that true? Let's just keep going, and hopefully it'll all fill in. We're in the inner ear, we're in the cochlea, we're trying to hear. We have to conduct sound waves, and you start by being able to have an actual replication of that frequency of that wave. It was at the tympanic membrane, and then it gets tapped out along the way in the ossicles, and then the stapes beats on the oval window, and it gets that fluid wave is conducted along the length of the cochlea. I think that's the sentence I was wanting to get to. So a sound wave becomes a fluid wave in the inner ear. And that's the first place, that's what I wanted to say. That's the first place that we're changing the nature of that wave, because it was a vibrational wave in air at the tympanic membrane. It's a vibrational wave in air in the ossicles, but this is where we switch to a fluid wave. And so we have this membrane that floats in there, and it's not floating, it's all tethered, and it's exactly where it's supposed to do. But it will receive that. and it sends, it will become very resonant to that wave at a certain point in growing. And then you can conduct that as an electrical impulse. So let's get to that. Shall we? Have a drink of green tea?
Does it become fluid already at the basilar membrane, or after it crosses a certain point over there?
Let's get back. I think this is where I feel like I missed something that makes this make more sense. Okay, so look at. Is this a good picture to see this in? Okay, yes. Tympanic membrane. So you see the outer ear. There's the tympanic membrane, and then you see the little yellow. I'm looking at this lower picture here, and so you see the 3 little ossicles. Those are also in air. That's the middle ear. And so when you get to this, all the blue bit, you're seeing the membranous labyrinth. It is filled with fluid, and it is surrounded by fluid. And the only place where that this is the part I didn't say that this makes more sense. So that membranous labyrinth is completely surrounded by bone, bony structure, except for this little window, the oval window. And that's the only place. Oh, there's another little place. I'll get to that. So that little oval window is where the stapes is interacting. So really, you get through all that noise in the world, and you're down to a little oval window. Is the only place that the world is in contact with the inner ear. And so, oh my gosh, I'm just thinking of this. It's making so much sense right now. I love it when I learn. You know, that really makes the inner ear like a soundproof booth. It really, I'm gonna get a little nerve chilly, y'all. It really does. All of the membranous labyrinth, where all the vestibular and auditory information is completely locked away, truly, inside this membranous bit. And the only place it connects to the outer world is at the oval window. Is that more clear? Okay. There is also another little window below it, inferior to it, called the round window. And that's also another little spot or portal between the inner ear and the middle ear. And that really is going to, this is cute, when those waves are running through there, they dampen, the wave movement dampens or ceases when you get to that part of the basilar membrane that receives it. But some waves will continue and can keep spiraling all the way through all those chambers, and you have to dissipate that wave energy. And so the round window can kind of bulge to dampen that. Isn't that cute, though? So it isn't like ripples in a pond that can't just keep going. They really have to stop or you'd screw this whole deal up. So the round window exists to just kind of help dissipate remaining wave energy. That was well said, y'all. I hope you're writing this down. So that's weird, though, isn't it? This is really physics, and this is why you go, I don't want to hear it, unless you don't because you love physics. But there are portions in every single thing you study that you resist, right? Not because it's difficult, it's because you don't want to hear it, because you can't understand it. And I feel like, and I don't know if you recognize this, but I certainly recognize this about learning because I continue to learn things and to teach things and whatever, so it's good to know how to learn and how you learn. But when you resist something, you can't learn it. And so the best thing to do is just drop your arms, girl, and just don't resist. You know, just drop your arms. And don't resist. And I can think specifically, you can think specifically about subjects. I don't know why I'm doing, no, I know why I'm doing this right now. Yeah, I do. You can think specifically about subjects where you just went, and you just close it off. That's okay. You don't have to learn everything. But the important thing to do, I think, in terms of your nerves and learning is if you're going to resist something and not learn it, then drop it. Forget it. Don't let it stress you out. miss those questions. Or you can not learn it, stress out, take away from what you're willing to learn, and miss all the questions. You see what I'm saying? So, or you can go, okay, I'm not willing to resist that, I think I'll learn it. And I remember specifically doing this about lipids. For some reason, fatty acid metabolism was one of those things I just blocked out and said, I'm not gonna learn that. I don't get it, I don't like it, they're insoluble, I'm non-flexible. I wouldn't learn it. But the minute, I was willing to just learn it, every single damn thing made more sense, So, which brings me to, this is like physics. So when you hear wave and frequency and amplitude and resonance and dissipation of energy, this may be one of those areas where you just go, I ain't gonna do it. Then don't do it. And that's okay. But understand that you'll miss those questions, but move on and put your energy elsewhere. And I think this is a great way to approach studying, Do you know what I mean, though? Because you have such a mountain to climb every time you sit down to look at information. You can't know it all. And take your areas, or what can happen is you go, this is a really difficult area. I'm going to dive into it and spend no time on the areas I'm really interested in. That's another way to go. And it should get you a D. Because the stuff that's really hard is really little. The stuff you already understand and love is probably really big. So anyway, you just kind of wipe things out. So I'm thinking you're blurring over because A, it's really late in the day. B, I'm just droning on. And then C, if there was some physics in there. So that's a lot of physiology. And if you don't resist physiology, and I'm not trying to sell you on physiology because you have to take it, you already bought it. That if you do take in the bits, or the big majority of the bits, all of the stuff makes sense. And one of the things that's going to make sense is the drugs. And I don't mean wait, let me rephrase that. What really will make sense is to take drugs. Now, the drugs that you use, that you will prescribe and you'll get people to use, which is going to be a huge part of your practice, You need to understand those, and they're all about physiology. Sorry, this is true. Now, they're basilar membrane, I'm writing a drug, no, but diseases that go about that, and a whole bunch of drugs have side effects in the inner ear. So, full circle, circled live, drumline, head cannon, basilar membrane, we're in the cochlea. The other thing I want to say is, when I sit down and make my questions, they have each person submit questions. And so I'll submit physiology questions. So you'll get 1 grade, you know this, for the concentration. And then there's another way to look at it, kind of linearly for physiology. And then all of those add up, so you have to get physiology passed and everything, and then all the vertical numbers passed, So the way they do that is they have me submit test grades, and they enter them into the program, and then they have ways of magically tagging them. They tag them so that it's physiology and different kinds of knowledge, and that's how it all works out. So they've asked me to submit my test questions, and how I do that is first I start out like writing on my notes, then I make all my slides, I make the outline, and then I write the questions. So the whole thing is done, and I just make sure there is a question for every section, for every single thing. But because I'm only asked to submit something, I think we're asked to submit like 12 questions per 2-hour chunk, something like that. That's not that many really, 10 to 12. So I have to cover everything. So one question may have some foils or choices that has a lot of words from other areas in it. So that's just how I produce them, so that they're very concise, they're very sciencey, because there's a lot of words in them that are terminology from this section. And so, the other thing is, what was the other thing I wanted to say about it? Did you ask me something about it? Did I answer it?
You said you were going to only present the information that was going to be on the
Oh,
test.
good, I'm glad you told me that. So, you have this textbook because I want you to have a reference, and I'm using it to order my material for all the stuff I'll be teaching, but I will never go to it and take something out that I didn't present. I've already ruled out what I don't want you to worry about from the book. So the book would only serve to kind of enhance or fill in gaps to a degree. So anything, I only use my notes as fodder for exam stuff, just so you know how that works. Anyway. Wow. I just killed a bunch of time. We're in the inner ear. We're conducting sound waves along the basal membrane, and it is resonating at a particular bounce or reckon or vibration where it likes that vibration the most. And its shape, its thickness, that all leads to that and why it works that way. And so it has all these fibers lined up horizontally along its length, and they are kind of tuned like strings of a guitar. So I don't know anything what you call a guitar part, but there's this part with the hole, and then there's that part with the strings, right? The strings here. And that's kind of how the basilar membrane works. So each of those little fibers or strings has its own little vibration. And so instead of being arranged like this along the length, they're horizontally. So you can have a lot of them. Instead of 5, you have, I wouldn't even know, a grillion. That's how many lined up, so that you can really recognize exquisite differences. Not just, not just a vibration, but exquisite differences, because that basilar membrane recognizes so specifically tiny little bits, tiny little vibratory bits. And a sound will have tons of different vibrations in it. So you're knocking on the oval window, but somehow that basilar membrane can read all the nuances in that. So, you're listening to music and there's all those parts and it's coming at you at once. And you only have this, bilateral tympanic membrane doing their thing, and the little ossicles doing their thing, and the punching on the oval window, and somehow all of that translates along the length of that fluid wave, along the length of that basilar membrane, to nuances in that sound, and tone, and pitch. Okay, but that's not it. Just vibrating your basilar membrane ain't gonna do it. You have to send the signal. So let's send that signal. What just happened? Weren't we there?
You don't go back. We went backwards.
But I'm pushing the forward one. Oh no. Oh, I went backwards. I got excited, went backwards earlier. Was unaware of it, right? Basilar membrane. Here we are. Organ of Corti. The organ of Corti. Wow, we're getting really dork out, man. Okay. So let's go back to the scalomedia, shall we? That wedge-shaped business portion of the cochlea. So the basilar membrane is at the base or the floor of that duct. That duct is filled with endolymph. And then the organ of Corti is sitting there on top of the basilar membrane. And it's not just a flat thing, because remember, this is a belt of information that's going all the way up through the cochlea. So this is just continuous. So we're just looking at little of it. So look at the lower picture for a little second and see the basilar membrane there. So you have some longitudinal and kind of cuboidal cells out there. Those are supportive cells. And then you have the business cells, which are going to be the mechanoreceptors. So in the eyeball, they were photoreceptors because they're receiving light. These are mechanoreceptors because they're sensory for mechanical deformation. And so they're going to sense this resonance of the basilar membrane, that basilar membrane moving. They're going to deform. So it's not the same as a chemical deformation. It's a physical receptor deformation. So those are called hair cells. And so there are outer hair cells and there about 3 of those to 1 inner hair cell. And look at the inner hair cell, you can see nerve fiber coming up, and there's the inner hair cell. So there are more outer hair cells than inner hair cells. But the hair cells are the thing that then the basilar membrane moves, and it deforms them, and then they generate an impulse to this nerve fiber. I had something I wrote, I don't recall, about hair cells. I don't see it here. When these hair cells get damaged, that's the most common cause of sensorineural hearing loss. So there's conductive hearing loss and there's nerve loss. And really, conductive is you can't get the sound waves in there. If you lose hair cells, you can lose that frequency. So it kind of depends. And it's natural to lose certain frequencies with aging. So it's really a loss of hair cells. Okay, let's look at that. Yeah.
How does the basilar membrane actually move the hair cells? Is it moving the cells on the bottom that moves the hair cells at the top?
It
Is the
physically,
resonation?
yeah, it physically bumps them. I mean, it's actually receiving the energy of that wave, and it will physically lift them. It lifts them, and they kind of bump into, you see this, oh, it's just perfect timing. This tectorial membrane here is a membrane that the hair cells are really touching. And so that kind of weights the situation down. So the basilar membrane will lift that up, but the tectorial membrane is going to help dampen and help the hair cell receive that, you know, as long as it's floating around. You have to have a real defined deformation. And hair cells can go one way or the other way. And one way hyperpolarizes them, and one way depolarizes them. So it helps kind of keep all that weighted and working. Hair cell, they are mechanoreceptors, meaning mechanical movement is what causes their excitation. They will synapse with cochlear nerve endings, and that will also be called, in that region, actually specifically called spiral ganglion. So if you see spiral ganglion, that still really means cochlear nerve endings. But we just have little chunks along the way before it exits, and it's really called cochlear nerve. The hair cell has stereocilia on it, which are then the hairs. This is where it gets its name. So instead of photoopsins, their thing that's receiving the stimulus is going to be the hair cells located on the surface of these hairs. They're located on the surface of the hair cell. And really, look how they extend up into the tectorial membrane. And then there's just some pillar, see this little rod of Corti? There's just some architecture that keeps all of this supported and separated and working, because otherwise you're talking about membranes and fluid. Things could get crazy. So you have a lot of ultrastructure. And so that's really the things to say there. So that's the hair cells with their stereocilia. Yeah, go ahead.
Will the sound in this region be fluid as well? Or
all of this is in fluid. Once you're in the inner ear, everything is in fluid. So yes, and it's the basilar movement that's receiving a fluid wave and moving and deforming upward and causing these hair cells to go, oh, and that gets them excited. You bump into them, it gets them excited. So again, a little bit more. Hair cells, there are inner and outer ones. This tectorial membrane, you see it there. And so see now, instead of just a line, a linear two-dimensional thing, you kind of see three-dimensionally. But that's how it's laid out all the way around the 2.5 turns of the cochlea. Let's see what I want to say. that's how it's arranged and that's how it works. The stereocilia, their actual hairs, are embedded in that pectoral membrane. And this is what we'll see throughout the inner ear. You're thinking, oh dear God, is this never going to end? No, it's not. Okay, so here, let's excite a hair cell. The top picture is no movement, and then look, the basilar membrane is in. And then that makes the hair cells shift because you've embedded them and you have them stabilized. When things shift, then it is a definitive, specific mechanical deformation that can cause the change. So it's really this, boom, now you can hear. But you're hearing at that certain frequency. So that information is sent as specific information along the cochlear nerve. That's really all it is. And see, it isn't so much the hair moving, I meant the cell moving, but the hairs themselves are just shifting. Those little cilia are sticking out, they're arranged in a particular kind of arrangement. They are actin filled. Actin, one of the contractile myofilaments, a protein, and it has myosin wrapping around there. We'll talk about how actin and myosin interact more later, specifically in cardiac section first. But what happens is, how can you have a hair cell that responds to deformation? You have protein ultrastructure in there, and these happen to be contractile, mobile kind of proteins that will conduct a signal once they interact. And so that intracellular signal will happen. Gates will open, neurotransmitters will secrete. So, movement of basilar membrane rocks and excites the hair cells. You kind of want to say rocks and excites hair cells, don't you? Because you're bored? No? Okay, just me. Now, let's keep exciting a hair cell. Look, here's the little stereocilia. Oh, God, this looks like it gets really complicated, but don't freak on me. So here's just a picture up there, like a scanning electron micrograph, where you see how they're arranged together around the central point. So what that is really doing is this collection of these things, and you want to be able to conduct the deformation, so if any of them shifts, it kind of gets sent around that whole group, so that they are sensitive. That a little bit of noise and a little bit of shift in that basilar membrane will recruit the entire cell to get excited, because you have plenty of mechanical change there. So these little guys are actually kind of connected together, and when they slide or move apart, it really makes channels open. So mechanically sensitive cation channels just means that positive cations will flow through them. Positive cations is redundant. Positive ions will flow through those channels when the hair cell moves. So depolarization, in this case, excitation of that cell occurs when these cation channels are opened. The cations that enter will be potassium, also calcium. Enter through these chambers because things have moved, these channels have opened, the interior of that cell becomes very positive, and so that's the wave of depolarization that will be transmitted along the remainder of that hair cell. From the stereocilia to the interior of that cell, the polarity changes, and that's depolarization. That's all polarization is: depolarization, hyperpolarization, repolarization, resting potential, action potential. All those things is you have polarity across a membrane. You have two fluids and a membrane, and one fluid's positive and one fluid's negative. It's polar, and that's all it means. And if they switch, then you depolarized it. When you set that back up, you repolarized it. So it's just polarity, positive and negative, and that's all it is. That's all electricity is just positive and negative charges. And the shifting of those charges is on or off. In this case, we're shifting the cell on by allowing a whole bunch of positives in. that depolarizes the cell.
So those two things together are the two hair cells? When they
That's,
rub
you're right.
against each other?
well, that's specifically showing the stereocilia of a hair cell. So they would be like located, kind of wedged into that tectorial membrane. So it's showing how they're together, but when they move, how does that happen? They're actually connected. There's a little actin and myosin activity that goes on, and it will physically pull open channels, which is so cute. Isn't that so cute though, really? You just open a gate, and the gate's open, and the cation's split in. So it really is, that's different than a chemical receptor. It's a mechanical receptor. Super cute. And then you repolarize or set that original polarity back up by pumping those cations out. Until it's repolarized, deformation isn't going to change anything to any degree to allow a signal to be sent. Also very exciting. Do you want to take a little breaky? Because we're going to finish. We're going to get through all of these slides, and then I'm not coming back till March. Yay! Aren't you excited? You won't have another 4-hour physiology lecture again. This is long. Do you want to take a little breaky? You need to take a little break. Go stand outside in your power stance.